This is a production of Cornell University.
Thanks, Rebecca. Thank you. Appreciate the very nice introduction. All right, let me get my laser pointer here. All right. So again, thank you, Rebecca. The work I'm going to present is my favorite of my whole career. I've really enjoyed being involved in working with an underutilized species, and I'm having a lot of fun with our community science project. So I'm happy to have this opportunity today to tell you about how we're blending both of those. This is an overview of the points I'll cover during my presentation. I'm going to start out with some acknowledgements and tell you about our Physalis team. I'll then move on to tell you about the evolution of the project. How did we get started working with the Physalis? I'll then move on and talk about characteristics that need improvement. These are being driven by priorities that we set in our NSF proposal, but also being generated from feedback from our community science group. And our community science project involves farmers, home gardeners, and various organizations. And there's two reasons for this. Is first, we want to get feedback on the characteristics that need improvement, getting that firsthand information. But also we want to familiarize people with, what are groundcherry and goldenberry, because I know a lot of people either haven't heard of them and certainly haven't had them. And then at the end, I'll move on to talk a bit about the research and how we're fast tracking that improvement, how we're generating enhanced germplasm with using gene editing. I'm not going to go into a lot of detail on this. I gave a talk about this in February of last year in plant breeding. So this is just going to be an update on some new developments. So this is our Physalis team. This is our core group. This is Esperanza Shenstone. Esperanza headed up our community science project. This is Linnell Randall, Carrie Swartwood, and Nathan Ream. We're standing in our field from last year, right after we got done planting it. We have some undergraduates in the group. This is McKenna Raspantini. You'll see McKenna is using our backpack shop vac to vacuum up all the dropped ground cherries in our greenhouse, making quicker work of it than what we used to use, a broom and a dustpan. McKenna is an undergrad at TC3, Tompkins County Community College. And this is Elliot Meyer. Some of you might know Elliot. Elliot is an undergrad at Cornell, and he's been doing a research for credit project in our lab. This is Savannah Dale. Savannah is a graduate student from Cornell who just joined our group this year. And this is Julie Davis. Julie is actually a graduate student in entomology in Jennifer Thaler's group. And Jennifer is working with Tomatillo, which is also a member of the Physalis. And she's been helping us with some of our community science. And also we're collaborating with Julie and Jennifer on an insect interaction project. So how did we get started working with the Physalis? Well, several years ago, we were working with tomato, and my collaborators, Zach Lippman at the Cold Spring Harbor Lab and Mike Schatz at Johns Hopkins University, were looking at the genes and networks involved in plant growth habit, fruit characteristics, inflorescence architecture in tomato. And we're using primarily a reverse genetics approach, so knocking out gene expression using CRISPR-Cas to see how it affected the phenotype. And while we were doing this work, we thought, wow, CRISPR-Cas could be really a powerful tool for fast tracking improvement of an underutilized species and creating this enhanced germplasm that breeders could tap into to make those improvements. So for our second round of funding, one of our aims was to take what we learned in tomato and apply that to a more distantly related species, and one that was certainly underutilized and had very little, if any, improvement. We decided to stay in the Solanaceae family, and this shows a phylogenetic tree of just some of the Solanaceae, and we chose to work with the Physalis, and the Physalis that you might be most familiar with is Tomatillo. But we decided to work with groundcherry and goldenberry, and this is for two reasons, because first we felt that this would be a great platform for answering our basic fundamental research questions, looking at the genes and networks that we found in tomato, but seeing how they can improve groundcherry and goldenberry. But we also thought there was potential for these fruit as a specialty fruit crop in the US, but definitely after some improvement. Now, groundcherry you'll sometimes find in farmers markets, and goldenberries you'll sometimes find in grocery stores. And people often ask me, you know, what do they taste like? And I think they're expecting them to taste like cherry tomatoes, but they're much sweeter than cherry tomatoes. And there's a difference between the ground cherry and goldenberry flavors. So ground cherry is sweet. We have one accession from Poland that has a slight pineapple flavor, but goldenberry is a much more complex flavor. It's this sweet, tart, tropical flavor. We have some that have kind of underlying flavors of mango, apricot, and coconut. And kind of a funny story to share. I recently gave someone some goldenberries. They had ground cherry before, and I gave him some goldenberry fruit from the greenhouse, and his reaction was kapow. He just wasn't expecting that burst of flavor from the goldenberry. And it's been fun because the different accessions we have these different flavors. So some characteristics of interest to us, it was that they were underutilized crops in the US. They're more popular in countries such as China, Germany, and Colombia. And actually, Colombia is the major producer of goldenberry. And this is a picture I took at Tops Markets here in Ithaca a couple of years ago that shows goldenberry in with the other berries like raspberries and blueberries. So these are grown in Colombia, but they're distributed by a company in Florida. They've got good nutritional content, vitamin C, beta carotene. They also have anti-inflammatory, antioxidant properties. And the physalis in general are known for their medicinal properties. And this is certainly true for ground cherry and goldenberry. They've been used over the centuries to treat different ailments. And they also contain these compounds for plant protection, these withanolides and physalins, which have steroidal properties. So these are just some of the germplasm, some of the sources that we've gotten our germplasm over the years. This is not an exhaustive list, just an example. And those that are in these yellow boxes are ones that we've worked with the most over the past several years. As for characteristics that need improvement, I mentioned that some of this is being driven by priorities in our NSF project, but also feedback that we're getting from our community scientists. So this is just a list of some of the improvements that we're hoping to make. Both ground cherry and goldenberry have this sprawling, unmanageable growth habit, requires supports and trellising, which you could imagine on a larger agricultural scale is going to involve a lot of labor. For ground cherry, it's got a small marble-sized fruit, so we would like to increase the size of that ground cherry fruit. Also, ground cherry has this fruit drop problem, and that's why it's called ground cherry. It often drops to the ground before it's fully ripe, and it makes it difficult with harvesting, but also there is a potential foodborne illness issue with gathering the fruit up from the ground and it gets dirty. And just to note that goldenberry does not drop its fruit. Ground cherry does, but not goldenberry. And then there's the husk that surrounds the fruit. And this can be, again, a labor issue with having to remove the husk for large-scale production of juices, jams, and other fruit products that they're used for. And one of the problems with goldenberry is that it's not as productive as ground cherry. It doesn't produce as much fruit, and it needs a longer growing season. That's especially true here in the Northeast. So this shows some plants from our field last year. This is about mid-season, so sometime in July. These metal stakes are about 7 feet tall. We've sunk them in the ground about 1 foot. But you can see how large these plants are. I mean, by the end of the season, these plants are to the top of these stakes. We also have to do trellising with some twine that we interweave in between the plants. And, excuse me, in between the plants, and that's called Florida weave. And, but again, you can imagine on a larger scale, this requires a lot of labor. This shows the plants at the end near the end of the season, and you can see how tall they've grown to the top of those stakes. These are ground cherries with the goldenberries in the background, and you can see that they don't grow as tall, but they're sprawling, and we still have to stake and trellis them. So again, it's a lot of work. This shows the fruit size. You can see the difference between the goldenberry fruit size here. This is from South Africa, and this is from India. Compared to the ground cherry fruit size, which is much smaller. This is an accession from the Netherlands, excuse me, and these are from Poland. Sorry. And this shows the real, I mentioned that we've got this problem with the fruit drop, and this shows one of our greenhouses after we've harvested fruit. Excuse me. So we so we use our backpack shop vac, but you can imagine on a larger agricultural scale. Excuse me, I don't know what's going on. It's pretty dry in my office, I guess. Farmers would have a real problem with harvesting these. So the reason for that fruit drop is this little joint here. It's kind of like an elbow, and the fruit drops in this region. And what we're doing right now is we're studying the biology of that region to try to determine, how can we fix that problem. We have tried crossing with goldenberry, but I think there's too much of a species divergence for it to be successful at the crosses. So, how did our community science project come about? Well, I felt that if we were thinking about these as a specialty fruit crop and doing improvements, I really wanted to hear from stakeholders, from farmers and growers firsthand early in the project to help guide us through that improvement. But I also felt it was important to heighten the visibility of Physalis as a specialty crop because, we didn't want to go through all this improvement and then spring the fruit on people and have them say, what is this? So I felt it was really important to have this educational component as well. So in 2018, I submitted a proposal to the Triad Foundation, and that's a local foundation here in Ithaca. The proposal got funded. I was very excited to have the funding to be able to do this. I decided to keep it simple and stay within New York State. And we had 11 farms that were involved, and each of these little red balloons here indicates the location of a farm. We provided seeds from 3 different types of ground cherry and 3 different types of goldenberry. And in addition to our growers and farmers, we also had 40 participants that represented home gardeners, public gardens, and we had some plants at the Cornell Botanic Gardens and also at the Ithaca Children's Garden summer camp. We gave people surveys for feedback. We visited the farms. To facilitate communication, we had a Facebook page, a web page, and a blog. And at the end, I really wanted to have a fun event and bring participants together. So we organized this harvest celebration event where people came to share their experiences, and we had some foods made with the ground cherry and goldenberry, and it really was a lot of fun. So this shows some images from some of the farm visits that we made. We gave the farmers some guidance on how to grow the plants, like how to germinate the seeds and the distance they needed to be spaced. But we felt that since we weren't paying them to do this, they were doing this just out of the goodness, the kindness of their hearts. We wanted them to adopt it into their production systems. So as you can see, this farm here, they put down black plastic. This is a high tunnel operation. This is another farm where Esperanza visited with the farmer and an extension agent. And you can see that these plants are absolutely gorgeous. They did an amazing job, and we got some really helpful feedback from our farmers and growers. So our farms represented farm to table, community supported agriculture, farmers who sold their fruits and vegetables at farmers markets, and also wholesale operations. There was one farm in Western New York that grew hundreds of pounds of ground cherries. Now that's a lot of ground cherries when you see how small they are, but they give them to this they sell them to this brewing company, and this company makes a cherry obscura, which is a sour ale beer. I have yet to get my hands on this, but one of these days I will, and I'm really dying to give it a try. So for community engagement, again, we had home gardeners and public gardens. I absolutely love the fact that kids liked growing these plants, and they also enjoyed eating them. Kids are tough, and I figured if kids really like them, then we're on the right track for a fun fruit for them. But it's not only the flavor. They enjoy opening them up and seeing this little fruit inside, which is just part of the experience of eating the ground cherry and goldenberry. We had, this is signage at the Cornell Botanic Gardens. This was in the Heritage Garden, if you're familiar with that. And this is Esperanza and our summer intern, Kyle Keating, visiting with Ithaca Children's Garden summer campers. So they were responsible for growing the plants, taking data, and they shared that data with information with Esperanza and Kyle. So this was, before 2018. Actually, we had only grown these plants in the greenhouse. So this was good firsthand information for how they would do out in a field setting. So I'm going to start from the bottom here. We had a couple of reports from home gardeners about possible blight and wilt, but really no disease problems. Chipmunks were the biggest problem for me in my garden. I never can seem to get a ripe goldenberry or ground cherry. But where we're really seeing, we saw some good observations and informations were with two insects, the three-line potato beetle and this Chlamydia subflexa. And it wasn't only that we learned about problems with these, but we also learned about some really interesting interactions and comparisons. So for the three-line potato beetle, this is the adult and these are the larvae. So the adults visited the ground cherry and goldenberry. They laid eggs on both, but where we saw the difference was in larval development and larval behavior. So on ground cherries, the eggs would hatch often, not as many, but we definitely saw an effect on the larvae. They either didn't develop well or only very few did, but they went to town on the goldenberry. We saw a huge number of larvae on the goldenberry. They did a lot of damage, and really the ground cherry were untouched. Kind of similar for Chlamydia subflexa, which is a specialist insect on the Physalis. We saw more of a problem with goldenberry than we did with ground cherry. So this shows the larval stage of the Chlamydia. And this is an image from one of the farms. This is in Poughkeepsie, New York, and it shows one of the stages of the larvae in this kind of obliterated fruit from the insect. So, and some comments that we got, I could fill slides with comments that we received from people. I just loved getting all the information from them. So for ground cherry, excuse me, this is a comment from one of the growers. He said that if you could get these plants to behave, meaning no fruit drop and less sprawling, we'd be into acres of these. So this is the first time that they had grown these ground cherry. And then I also, excuse me, have had a few people ask when they were going to get the gene edited lines. When would they be available? For instance, we had one organic farmer here in Ithaca who wasn't interested in growing ground cherry again because he said he had grown them in the past and they were just too much work, but he'd be happy to give the gene edited lines a try, which was great to hear. And another story to share is that there is a project participant in Minnesota. Her name is Betty. And Betty doesn't have internet access. She doesn't do online, but she's called me a number of times over the years. She found out about the project through an article in a magazine or newspaper. And she called me one day earlier this year and said, now, Joyce, the seeds you gave us, they're not from the gene edited material, the gene edited plants. And I said, oh, and I thought, oh my goodness, she's concerned that we have given them gene edited material. I said, oh, no, Betty, these are all the unimproved lines. We just want feedback from people to know how we can improve them. And she said, oh, I really want to give the gene edited lines a try because I've grown ground cherry for many years and they're really a problem. So whenever the gene edited lines are available, please share them with me. So that was, it was really good to hear that there was not too much concern on Betty's part about having these gene edited lines. As far as flavor, we got some good feedback on flavor. And this one person said that this is GB, means goldenberry, was amazing and very different from ground cherry. And again, this is these last two comments point to the problems with the beetles. This one person said that the potato beetles definitely prefer goldenberries, which substantiates some of the other observations that we got. And one of my favorite comments was this one, I have never fought so hard for a crop. And the three-line potato beetles were relentless. So again, really good feedback for us that, originally when we put in our NSF project, we're thinking about agronomic characteristics such as plant growth habit and fruit size, but we started to learn about insect problems as well from this first year of our community science project. This is a screenshot of our website and blog. If you're interested in taking a look, it's called the Physalis Improvement Project. So if you do a search for that, you'll find it. You can subscribe to the blog. And we also have Twitter accounts. I post information often about the project on my Twitter account, and then we have this Physalis project account as well. So for our harvest celebration event, we invited participants. We had foods that were prepared with the Physalis. We had we had a Groundcherry rice pudding. We had these little muffins that had ground cherries on the top. I made a granola with dried golden berries that I got the dried golden berries off Amazon. We also asked people to share their experiences with growing it. Every event that we do, we take our whiteboard and some Post-it notes to ask people to come up with the name for the ground cherry, because when we fix that fruit drop problem, we're not going to call it ground cherry anymore. So these were 2 suggestions. We also had activities for kids, and we gave these mugs out as thank you gifts. So it was a great event. We had a lot of fun, and we decided that we would do this again in the following year. So in 2019, I had some carryover funding from the Triad, and I used that preliminary information from the Triad to apply for an NSF supplement, and I got that. So we felt that since we were a success in New York State, that we could expand into other states. So we ended up with 70 participants in 18 different states, indicated by each of these blue balloons. This year, we made some connections with chefs. We took fruit for chefs to be able to have them come up with some dishes, because we felt that if chefs wanted it, then they would ask farmers to grow them. And this was a great outreach for consumers who maybe had never heard or tried Groundcherry or Goldenberry. We also did a consumer panel evaluation that I'll tell you about on the next couple of slides. Julie went to the farmer's market to do a taste testing. And Esperanza and McKenna again visited with our next group of summer campers at the Good Children's Garden. So for consumer panel evaluation, we did this in collaboration with Cornell Sensory Evaluation Center right here on campus. We had to limit it to 100 participants. We ended up with 102, and they each got $5. This shows McKenna and Esperanza in the kitchen of the evaluation center. You can see all the fruit they prepared, and then there are these metal chutes here that they put the fruit on a tray and pass it to the participants on the other side. Each different type of fruit had a number associated with it, so it was a lot of work in preparing this. The evaluation center personnel told us to keep it simple, so we had just 2 types of ground cherry and 2 types of goldenberry. There were 22 questions for the participants to answer, with 11 qualifiers like, I like it extremely, I like it very much, etc. We also had a comment section. In addition to flavor, we asked people if they would like it with or without the husk, and we got some information about color and size preferences, and also about seediness. And there were these additional questions that we asked people to complete. And after all this, I was thinking, wow, that's a lot to ask people. I wonder if paying them $5 is really enough. I mean, I certainly felt, especially after seeing the results, that they deserved more than $5. So this shows right here down in the bottom right-hand corner, the participants on the other side of the kitchen. They are answering the questions on the computer. When they need the next round of fruit, there's a little button they can push, and then Esperanza would put the fruit on the tray and feed it through to the participants. This is great, really, to see behind the scenes of how this taste testing, these evaluations are done. So we got a ton of data back, but I'm just including some quick takeaways here. So 53% had never tried ground cherry or goldenberry, so a great way to familiarize people about what they are. We found that 75% preferred the ground cherry flavor over goldenberry, and then there were some preferences for within the goldenberry itself. Now, Columbia is the one that we got from the Tops Market and saved the seeds and grew those up in the field. We knew that they really didn't taste all that great, so it was we wanted to see what, we didn't think so, we wanted to see what consumers thought, and they agreed. So I was really happy to hear that 82% of the participants would actually purchase them. An interesting result was that 60% of people prefer to have it with the husk, and the comments were that it was more natural, more closer to what the fruit, actually was naturally, and it was fun to open up the husk and see the fruit. But there were some concerns that, do we know, are we going to find mold or insect in the fruit, which does happen sometimes. And something that I was really surprised about was that seediness was not a problem. There are a lot of seeds in these fruit, and I thought people might have an issue with it, but they didn't. As far as size, these are goldenberry and ground cherry. We found overall that people felt that the ground cherry was too small, but India was too big. Colombia was just right. So now we know for our improvement purposes is to really get somewhere close or right on with the Colombia size. We also got some feedback on color, and we're using all of this information now from the consumer evaluation, again, to help guide our improvements of ground cherry and goldenberry. So this year, we got close to 1,400 seed requests. I'll talk a little bit more about that in the next slide. But basically, this was a result of an article in the Los Angeles Times and the LA Times. I gave one of the writers talked to me about the project, and we ended up getting a huge response. We had to cap it at 750 participants because of the amount of seeds we had. We ended up with 43 different states represented, and my goal was to have somebody in Alaska and Hawaii, and we actually had some participants in those 2 states as well. So I'm really, really happy to see that we got really great buy-in to the project. In addition to individuals, we also had various organizations involved in the project this year. And some of the comments we got back was that it was great to have a project like this during this COVID, during the pandemic, to kind of help keep people's minds off things for a while and to have something interesting to do and to participate in a project. So going back to that article in the LA Times, an online version of the story came out in early January, and we ended up with a lot of requests at that point. And then the editors liked the story so much that they asked if they could put it in print, and so they did. So we had another article come out in print. This was in February of this year. So Esperanza was getting really worried, especially after the online article. She took the request and graphed it. So here's showing the graph versus months versus number of participants. And you can see before the article came out, we were around 150 participants. And then after the online article came out, we were up in the 600s. Again, Esperanza was worried about the print version coming out. And I said to her, oh, don't worry. Who reads print versions of newspapers these days? All of our requests are going to come from online. Well, I was wrong, because we ended up getting a huge spike in requests after the print version came out. We had to shut down the request site, and our business office told me that the phones ended up burning, as they said, with people calling to get seeds. Esperanza and I each got emails, and I even after that got letters from people asking to be part of the project. So really it was overwhelming to see how people wanted to be involved and wanted to help out with the project. So this just shows one container of envelopes that we sent out seeds, and we ended up with over 800 subscribers to our project blog. So a great way to be able to educate people about ground cherry and goldenberry. We're using iNaturalist this year to help gather all that information. We're focusing on the 3-line potato beetle, but we've also asked people to take pictures and include information of other insects or just of their plants in general. So the way this works is they can take a picture, it comes with an identifier. This shows a 3-line potato beetle. We can look at the observations, we can look at the pictures, and we can also graph. This isn't our data from this year. This is just some other data that Esperanza included in the slide to show an example of how we can graph all of that data. So the project iNaturalist ends the end of this month, and then we'll go in and take a look and graph all that data. So again, our primary information we wanted was on insects, but we also asked people to gather information on other characteristics as we did in previous years. We included a blog series this year on crop improvement. So each time that we have a posting, it's about a different aspect of crop improvement. So we started out with plant breeding, We had one on mutation breeding, and this is this is a more recent one that Savannah Dale put together on genome sequencing and how that's part of crop improvement. We'll eventually work our way through getting up to gene editing as well. I really wanted to expand connections with chefs this year, but because of COVID, that made it really difficult, almost impossible. But I really wanted to show the public what could be done with these. So I thought, oh, I know Zoe Dubrow in plant pathology, and Zoe is an amazing baker. I follow her on Twitter, and I saw some of the just gorgeous desserts that she would make and post. So I asked Zoe if she was interested in giving our ground cherries a try. We had a lot of ground cherries at that time, earlier in the season, and if she would write a blog for us about what she created. So you can see this here is a tart that she made with ground cherry curd. It's absolutely beautiful. She made these, they're a French little cake that she learned about when she was doing some research in France, in plants, not in baking, but she learned about these. I think they're called financiers. I probably have slaughtered that name, but she put the ground cherry in that and had the husk up at the top here. She also dried the ground cherries and made scones and used that ground cherry curd. She posted this information on Instagram. She got a lot of hits. I followed some of the comments, people asking where they could get ground cherry and goldenberry. And if you're interested, go to our website. Zoe's got links to her recipes on that post. And we also have recipes on that site for some of the dishes that were part of our harvest event. So to summarize this part of the project and where are we going, this has been, as I mentioned earlier, so much fun, and I'm just so happy to see how people really want to help us. I need to write proposals, get more funding. We're kind of on a pause right now because agencies that I wanted to apply for funding for outreach type initiatives were not taking proposals this year. I had funding for Esperanza only until June of this year. She's moved on to another position. So I really need to get some more funding to get somebody in to help with this. In the future, I want to expand our number of farm to table and CSAs, connect more with local chefs, and some other ideas that we might be able to come up with. So if anybody has any ideas of where I could apply for funding for some outreach and this kind of community science, I'd be happy to hear about it. So now I'm going to move on and just give you a bit of information about some of the lab work research that we're doing and how this is being guided by our community science information and also some of our own priorities. So we're fast tracking this improvement with gene editing. Part of it is that to create enhanced germplasm that has variability that breeders could tap into for improvement programs. But before we could do the gene editing, the first place we had to start was in developing a transformation method. And we started with ground cherry because ground cherry is a diploid, goldenberry is a tetraploid, and we felt with editing, the diploid would be a much easier one to tackle first. So we based this on our tomato transformation method. So we put ground cherry seeds in culture, they germinate. A difference between this and our tomato method is that for tomato, the cotyledons are the explant that works the best, but for ground cherry, we found hypocotyls actually regenerate plants that cotyledons do not. So that's a big difference between tomato and ground cherry. This is just our regeneration scheme. This isn't transformation. The medium contains growth regulators that induce this callousing in shoots. We then get plants that root. We take cuttings and they're put into a test tube. And the fully rooted plants are then evaluated for genotype, with their genotype to look for edits. When we have those rooted plants and they've been genotyped, if they're what we're looking for, they get transferred to soil. We cover them with these water bottles that have the bottoms cut off, help us gradually acclimate those plants to the real world because they've been in these very humid culture vessels. We take the caps off after a few days and then completely remove the bottle after about a week. And you can see how those plants transition really well. It's almost like it's 100% going from tissue culture to soil for us. This is the vector system we've been using. We started using this with tomato. It's got this cassette here with the NPT2 gene that confers resistance to the antibiotic kanamycin. We include the kanamycin in our culture medium to select for only those cells that received this vector. We have the Cas9 being driven by the 35S constitutive promoter, and our guide RNAs are driven by the Arabidopsis U6 polymerase promoter. Now you can see we use 2 guide RNAs. You really only need 1 guide RNA if it's working well to get edits. But because we were doing a reverse genetics approach, we really wanted to slam expression. So we were using 2 guide RNAs spaced about 50 to 100 base pairs apart. But over time, what we found is even though we follow all the rules for designing guide RNAs, sometimes one works and the other one doesn't. So by including 2 guide RNAs per gene, we actually can kind of save ourselves time. We then take this vector, we electroporate it into agrobacterium, and we do the transformation experiment. For our editing scheme, we edit those first lines, which I'm calling E0 instead of T0, which is actually often used for transgenic lines. But since this helps us separate what our overexpression transgenic lines are from our edited set. So we genotype those E0s for edits. We transfer them to the greenhouse. We then PCR that E1 generation for Cas9. We only want to move plants forward that are Cas9 negative, that have just the mutation or the edits. We then do the same for the E2 plants. We genotype them for edits. And all along here, we're looking at the phenotype, but it isn't until we get to the later generations where we start to take a deeper evaluation of the phenotype. Sometimes we have to go back to the drawing board and choose a different gene to edit. But then when they reach a point where we feel really good about that phenotype, we'll cross it with wild type, evaluate it some more, and genotype it as well. So again, just generating this, these edits to see what's going to make the difference and what we see as a good phenotype that carries through several generations. Now, as for the targeted traits of interest, again, this is just going to be brief compared to maybe other presentations where I would talk about the work. So I'm going to focus just on one gene that we've targeted for plant growth habit. There are a number of genes that we have tried, but this is the one that we have kind of the most history with. This is self-pruning, or SP. So self-pruning is a flowering repressor. And if you grow tomatoes, you know that there are these indeterminate types that grow really unmanageable and large. And then there are the determinate types. Well, the determinate types are because of this mutation in SP. And it was back in the late 1920s that a grower saw this compact tomato in his field, and over time it was determined that it was a mutation in this SP. They have this determinate growth habit, they have bursts of early flowering, and breeders took this material and started to put it into their commercial lines, and it really revolutionized commercial tomato production. Well, we found that counterpart in Groundcherry. This sequence up here is the wild type sequence, and these are what our guide RNAs look like. And then our this are just some of our edited lines, and you can see where those edits happened in both of those guide RNAs. This shows the phenotype of just one of our lines here. You can see this is the wild type, how tall it is compared to the edited lines. If you're interested in reading more about this and some of the other genes that we've edited for plant for different characteristics, we had a publication back in 2018. So again, following through, looking at the next generation and seeing if this is the type of gene that is going to give us what we want in later generations, or if we need to go back and look at something else. For fruit size, just like plant growth habit, there are a number of genes, but I'm going to tell you about this clavata1. So clavata1 works in concert with Wuschel to keep shoot meristem maintenance normal. And so you can imagine that a loss of function would cause an increase in the size of shoot and floral meristem. And the first sign that this was actually working in our Groundcherry lines was that in the wild type Groundcherry, the flowers have 5 petals, but in our CRISPR lines, we had up to 7 petals on those flowers. We saw a 24% increase in fruit weight in the edited lines. We also saw an increase in locule numbers. So locules are those little compartments that hold the seeds. Wild types had 2 locules, and the edited lines had 3 locules. The sequence down here, again, this is the wild type. It shows you what that sequence looked like. And as I mentioned before, sometimes one guide RNA works and the other one doesn't. Well, that was certainly true in this case for guide RNA 1. We didn't get any edits. The guide RNA2 did give us edits, and that was enough for us to be able to see a difference in fruit size in these lines. So 2 experiments that are at the early stage are those to address this fruit drop issue in abscission and also insect interactions. So this is work that's being done by Nathan Ream in the group. As I mentioned earlier, the fruit drops because of this little, this region here. We tried crossing with goldenberry, it didn't work. So we've done RNA-seq of this joint region at different stages of development, early, middle, and late. And right now, Nathan is plowing through all this data to try to determine some targets that we could try. Now, you know, some tomatoes do produce a joint, but they don't drop their fruit. So I'm imagining there is a different mechanism that's involved in the joint and abscission in tomato versus ground cherry. And also we did target some of the genes that have been reported and that we've seen responsible for joint in tomato, but they didn't work in the ground cherry. So, you know, this RNA-seq information is going to be really helpful for us to try to determine how to move forward. For the insect interactions, again, this came about because of our community science work. I mentioned those differences between ground cherry and goldenberry, that it appeared that the goldenberry was more affected by both the 3-line potato beetle and the Chloridia subflexa than the ground cherry. And actually the 3-line potato beetle preferred the Physalis over potato. It was the first time I grew ground cherry and goldenberry in my garden. Was the first time I had these beautiful looking potato plants, because all the 3-line potato beetles went to the ground cherry and goldenberry, but they definitely prefer the goldenberry. Again, I submitted a proposal to the Triad Foundation to get some preliminary information that we could use to go after a larger grant for this work. We started by doing an unbiased metabolic profiling of this material. Right now we've completed it for leaves. We're looking at husks and fruit as well. We're using LCMS, and we're doing 3 accessions each of ground cherry and goldenberry. This is work, again, being done by Nathan Ream, and we're collaborating with Frank Schroeder's group, and this is Bennett Fox, who is a postdoc in Frank's group. For the ground cherry, we're using this ZL05, which is a line we got from the Netherlands. GO is Goldie. It's one that's available through Johnny's Select Seeds, and this is the pineapple, which is one of our accessions from Poland, and the goldenberries is India, South Africa, and Colombia. So just in brief, for the method, we're using freeze-dried material that we then extract, and then we run that on the LCMS. These are 2 chromatograms showing the differences between ground cherry and goldenberry. I mean, what I want you to take from this is very visual and looking at the differences in the number of peaks and the height of the peaks. But please note that there are differences in scale here. So you can see there's definitely a difference in these metabolite profiles between the ground cherry and goldenberry. Nathan then took that information and generated this heat map. And the way we interpret this is that those in yellow are the most abundant, and each line represents a different metabolite feature. So you can see globally that with the ground cherry versus goldenberry, there are definitely differences in the types and the amount of metabolites. And there are also differences in between each different type within ground cherry and goldenberry. So we're taking this information, doing a deeper dive, and doing leaf bioassays as well in collaboration with Jennifer and Julie in entomology. So to summarize this part of the talk, CRISPR has been a really valuable tool. We were able to make those modifications that I mentioned for the SP and the clavata in just 2 years. We're moving into goldenberry. We have our first edited lines of goldenberry in the greenhouse. We're going to expand on that work with fruit drop and abscission. For our metabolites, we're focusing on with analytes right now because they've been reported to be involved in insect interactions in the Physalis. And we're doing some metabolic engineering to create mutants to help us tease apart which with analytes might be involved. And with that, I'd like to thank our farmers, community scientists, and public gardens. I mentioned my group earlier. Bennett and Fox involved in the metabolite work, our greenhouse group for all their patients with all that dropped fruit. Christoph, Jennifer, and Julie are involved in the insect and metabolite work. Zach and Mike I mentioned earlier, and our funding agencies who've made this all possible. And I want to thank you for attending, and I'm happy to take questions now.
Well, Joyce, thank you so much for a really wonderful journey through your work on Physalis. That was absolutely delightful and very impressive too.
Thank you.
A real tour de force from participatory research to successful editing and domestication. And can you please add me to your list of extremely large numbers of people who want seed? I enjoy, in my years in Peru, I really enjoyed the goldenberry. And even now, if I get a packet, I mean, a basket of either one of those crops at the farmer's market, I just eat it in one sitting, annoying my family members because I've eaten them by the time they get anyway. So yeah, look, questions, please. Any questions or comments? Isabel, are you there for a question? Okay, just being friendly, thank you. Who's got a question? Oh, let me look at the chat. We've got a few chats in here. Yeah, Brennan, can you go ahead?
Yeah, I guess I can ask it. Yeah, so what was the demographic distribution of like the people you engaged in your community science project? So, it sounds like you were able to recruit people across all different states, but sort of what was, the differences in like cultural backgrounds, living environments, incomes, just because it'd be interesting to see how that would vary across different demographics, right?
Yeah.
More groups are more engaged with this novel fruit than others.
Yeah, that's a great question. And it's not something we have that information on at this point. I mean, we were just kind of focusing on getting people involved and educating people, but it's not something that we have tracked. But I think it's a great suggestion for something that we should track going forward. For sure. So I'm not really sure how we might get that information now. If you have any suggestions on how we might do that, I mean, we have our map and we show the different locations, and we can go in and track where people are located, like where they, because we've got the information about where they live. But again, it's not something that we've tracked, but a good idea.
There's a couple of pieces of fan mail from Jean-Luc and Mark. So just FYI. Maybe to that, similar to Brennan's question, I would like to ask, is there some way in which the people in the testing network, your participatory network, are they kind of related to each other? Like, are they aware of what each other are saying? And could there at some point be questions about, what's our demographic structure and, who are we and, who's like you or who's not like just some way that you can socially network?
Yeah, one of our participants this year actually suggested, and I can't remember what it was, but she suggested an application that we could use to indeed do that sort of thing, to have people interact and get more of that information. And that's something that we would use going forward. I saved that email and we'll use it. I just can't remember what it was called.
Greg, go ahead. Gregory Vogel, got a question up there?
Yeah, thank you for the really interesting talk. I was wondering if you could talk a little bit about the reproductive biology of these Physalis species, and in particular, if they self-pollinate or outcross, and if there's any self-incompatibility.
They do self, there is a small, different species, not the ones that we're working with in general, but there are other Physalis species that do have some level of outcrossing that happens. And as far as incompatibility, we haven't, at least with the types that we've been working with Groundcherry and Goldenberry, we haven't had a problem. But we have done some work with Tomatillo, and some of the Tomatillos definitely have a self-incompatibility problem. So what we've done there is we've recommended that people, and there is some outcrossing with Tomatillo, we've recommended that people plant the, couple of different types and more than just one plant in order to get some fruit for those. So we would have to do crosses with the Tomatillo in the greenhouse to get fruit on some of them, but not all of them. It was just certain Tomatillos that we had a problem with.
Over to you, Isabel.
Yeah, so I do actually have a question. So I was wondering, in the sensory study that your lab conducted, were there any like commonly mentioned flavors that people like disliked? Just because I tried ground cherries a long time ago when I was a freshman, I think, and have since made a whole bunch of my friends try them because I've gotten them from the farmer's market. And a lot of them actually said that they got this weird tomato-like aftertaste that they weren't a huge fan of. But I wonder if that if any other flavors popped up.
Yeah, we had this really cool wheel that they generated with all the information that people talked about, different flavors that they had. This gorgeous color wheel that I didn't show today that helped us, like under, the chocolate or laundry detergent or, kind of other, some other things. And we so we kind of threw in that Columbia one as a ringer because, I don't know, there's probably some sort of agronomic reason why they've chosen to distribute that one, at least through Sunbelt, but it tastes awful. It's got this terrible aftertaste. It was my first time tasting goldenberry, and I was really excited, and I tasted it, I was like, oh, this is horrible. But since that time, we've gotten accessions from other places that are absolutely delicious, and they don't have that aftertaste as some do. So it really depends on the type that you're working with and what we see. We have one that is from Austria called Sherburne Gold, and it's got this apricot flavor, and it's just has none of that aftertaste that you might find in some of the other accessions, especially like the one from Colombia.
Thank you.
Sure. If you'd like me to share that wheel of all the flavors with you, I can share that slide if you shoot me an email if you're interested.
That would be awesome.
Yeah.
I took a couple of classes on coffee and cacao, and it would be really interesting to see one for fruit.
Oh, sure. I'll send you the whole presentation information. So
Thank you.
Cool. Well, if there are any other questions, step up now. If not, we're at time anyway. So I want to end by just maybe reading Jean-Luc's comment, which thanks for fantastic talk. Summarize my impression here. So thank you so much. That was really inspiring and fantastic. And I look forward to, Joyce, again, if you have any extra fruit or seeds, I'll volunteer I'm a terrible gardener, but I'd love to try on the allergy site. So, thank you so much. Thanks, everybody.
Bye. Bye.
This has been a production of Cornell University, on the web at cornell.edu.